Okay, good morning, everyone. Good morning. I'm going to try not to stand in front of the projector so I don't blind myself. How is everyone today? I know it's early. Yeah, I see some definite not morning people in here. Okay, so the first module today, we're going to be talking about life in the compost, and as an extension of that, life in the soil. So my name is Janel Luke. I am one of the trainers in the CCGS2 program. I was a site operator during CCGS1. I run a small compost operation for a nonprofit up in Paradise, California. And soil ecology and soil chemistry is pretty much my life. I won't bog you down too much with chemistry this morning. Again, it's early. But those are really my passions, so beyond this presentation today or any time in the future during this program, if you want to reach out and kind of go deeper into that stuff, you're welcome to. So a little bit about myself, aside from the composting. I moved out to California about 16 years ago. I'm originally from the East Coast, and I grew up in agriculture. So soil has been a big part of my life, even though I didn't really realize it when I was a kid, or at least didn't appreciate it. After I moved back out here to California in my early 20s, I started going back to school, and I was going to college for medical microbiology. I wanted to help people. The medical field felt like the best way to do that. So I spent a couple years going to school at a local community college, and then I spent a year in West Africa, in Ghana, and I studied pharmaceutical microbiology and interned for the West Africa AIDS Foundation. And it was during my time there, I was also involved in some farming in a local community. And when I got back from Ghana, I started thinking about the implications of environmental health and how that impacts human health. Obviously, we hear a lot about pollution, air pollution, water pollution, things like this affecting people, but even down to the biology in the soil and how that affects the food that we grow and literally affects the microbiome inside our bodies. The gears started turning. I read an incredible book during that time by a woman named Sonia Shea. It's called Pandemic. And her storyline in that book outlines how pathogens come into contact with humans through environmental degradation and destruction, which really inspired me. And I started getting into permaculture with some different local organizations. Shortly after that, literally months after getting back from Ghana, our town experienced the Camp Fire in 2018 and was completely leveled. And sorry, this is very fresh on my mind because we just had our 5-year anniversary on Wednesday. And it just really changed everyone's lives in our community. We started a nonprofit literally while the flames were still burning, and just wanted to find ways to help people. And we discovered that a lot of the questions that were coming out of it weren't questions about humanitarian aid, they were about the environment. Suddenly, all the people in this community were interested in what was happening in the soil, what was going to happen with the plant life and vegetation. So it really kind of opened up this opportunity for us to get involved, and we've been doing that for the last 5 years. We just had an event on Wednesday and gave away fruit trees and 1,000 pounds of compost in our local community. And people were really excited. So it's nice to, be in a group of people who see the value in all this. And I'm going to move on now, but that's a little bit about me and my interest in soil biology. So we're going to talk about life in the compost. So your compost is alive. Compost is a living cycle that we are creating to amend soils and grow plants and do other pretty exciting things that we can achieve with all of the biology we're creating in this environment. So there's lots of different ways that you can tell that your compost is alive. And I really like to start off by just encouraging people to have this sensory experience when they go to their compost lot. Because I have a bunch of fancy tools here. I am a tool person, I am a data person, but that doesn't work for everyone, and it doesn't have to work for everyone. So the same ways that we can tell if our compost is alive are some of the ways that we measure our own health and comfort and how well we're doing, right? So a lot of you have probably walked out to your compost yard or seen compost when it's kind of cold outside, and you see all this incredible steam coming off those piles. That is your compost pile breathing. All of the millions of microorganisms that are in your pile are respirating. So ideally, we have a collection of microorganisms that respirate in the same way we do. They're aerobes. They need oxygen to breathe in, and they breathe out CO2. Now, the way you can tell if your pile is breathing in a way that you want it to is that's not going to stink. There are other types of organisms, which we're going to talk about in a minute here, and the way that they breathe is quite a bit different, and the things that they're going to breathe out are going to be stinky, and they're not going to be nearly as warm. So seeing the steam coming off your piles is a good indication that you have a healthy, living compost pile. Another way that we can tell is heat. So this is kind of in tandem with that respiration going on. A compost pile should have a temperature. A healthy temperature, which I'm sure most of you know at this point, is somewhere between 130 and 160 degrees Fahrenheit. Slightly different than our body temperature, right? If you had a fever of 160, there'd be a problem. But ultimately, we can test the temperature of our compost pile and know that those microorganisms, they're breaking chemical bonds, they're consuming all of these materials that you've put into that pile, and their metabolism and respiration are what are causing the heat in your pile. It should not be the ambient temperature outside that's causing that heat. That's not how we keep our compost warm. And a great way that you can tell if your compost is hot enough is stick your hand in it. Can you hold your hand there? Because you shouldn't be able to. It should be quite uncomfortable within the first 8 to 12 inches of the inside of your compost pile. It should be pretty unpleasant. Another thing that your compost needs to live is moisture. We all need water to survive, and microorganisms are no different. They use that moisture not only to keep their body in balance and hold their structure, but they also use that moisture to move around to different parts of your pile. Without moisture, small organisms like bacteria and protozoa can't move around inside of that system. So in the soil, it's a little different because there are other organisms that might be present that can help those microbes move around a little bit. But in compost, it is super critical to keep that 50% moisture going so that those organisms can get to their different food sources. And texture. So this is something fun, and I had a volunteer offer to let us use their sample. So texture, surprisingly, is another way you can tell if your compost is alive. So you don't have to if you don't want to, but I encourage you all to put your fingers in this jar and just kind of smear around that material a little bit. So in healthy living compost, those organisms are breathing, they're eating, they're pooping, and they're moving around. And in doing all of those things, they're exuding different materials into that environment. And those materials have a very distinct texture. You'll notice when you touch that it's almost a little slimy feeling, but it'll roll up into a ball. It still kind of breaks apart. So those materials, organisms excrete to digest food outside of their bodies, and they also excrete them to glue themselves to each other, to glue themselves to food sources, so that they don't just get washed away when that soil moves, or when a heavy rain comes, they don't just get flushed away. And those materials are what create aggregation and texture in your soil. They start gluing together all of these different compounds, some of which they make, some of which are just organic matter in your compost. And they create these little granules that allow air and water to move through your materials. So I'll give you a second to pass that jar around. It's not a compost workshop if you don't leave with dirty hands, even if we're in a classroom. Yeah, so texture. And these are ways you can tell if your compost is alive and if it's doing well. All of these things should be happening. So these are the critters that are responsible for doing all of those things in your compost pile. This is the soil food web. There are a lot of different models you can find online that look like this, that may include different organisms. This, of course, is a very simplified chart of those different organisms, but I like this one because it also includes inorganic substrates in it, which isn't something you usually see. We see organic matter, which obviously this is all of the feedstocks that we are going to collect and put into our piles. Inorganic substances, this may include some types of contaminants that come in on your feedstocks, and a lot of my presentations have an underlying thread which is intended to give people hope that some of these other materials can still eventually be broken down. So anyway, primary producers, that's going to be all of your plant materials and things, organic substrates that you're collecting to put into your compost pile. And those are going to feed some of the most key organisms in your compost, which are really heavily responsible for the heat, the breakdown, and the accumulation of those materials that you're feeling when you touch that compost. And those are our herbivores and decomposers, fungi and bacteria. These are things that you're going to hear over and over and over and over again about as your composting journey continues. Fungi and bacteria are wholly responsible for the breakdown of different compounds in your substrates. They are also some of the driving forces behind the creation of soil. So your parent materials in your soil, your sand, your silt, and your clay, these organisms produce exoenzymes that break down those materials at their most basic chemical structure and can release nutrients that are present in those soils that were not available before. There have been some pretty incredible discoveries that even in soil that may not seem like there's any nutrients available in it, by adding biology back in, we can release nutrients, and it's an almost unending pool of material that exists there. So you'll never look at sand, silt, and clay the same way again. So in compost, same idea. They're taking those woody materials, that food waste, they're breaking them down into these really basic materials that are going to create what we call compost. So moving up the scale a little bit, the interesting thing about bacteria and fungi is as they're feeding on those organic materials, they're locking up that carbon and nitrogen in their bodies. Fungi are one of the primary materials in soil that make up our carbon pool. So fungi have a higher carbon need than bacteria, and bacteria are responsible for binding up nitrogen in our soils and in our compost. So we can't just have bacteria and fungi. We need these higher organisms. So the microorganisms we're going to talk about are going to be things like amoeba, flagellates. They fall into the class called protozoa, and they're predators. So just like in a forest food web, you think, okay, we have plants, and then we have animals that come and feed on those plants, and they poop, and they might release a little bit of nutrients, but then the predators come in and consume those organisms and continue to release that energy. So the protozoa are what are going to release nutrients from those bacteria by eating them, and they're going to continue that balance by putting nitrogen back out into the material in a plant available form. There's a bunch of different types of nematodes that also contribute to that part of the system, and even up to the point of predator nematodes that eat pretty much everything. Anything that can fit into their mouth, they will eat. The most important part about the soil food web is this needs to be balanced in our compost, and it needs to be balanced in our soils. Without that, we're not going to have plant available nutrients, and we're not going to have cycling of materials into and out of the soil, and we're going to talk about that a little bit more in a minute. Any questions? Yes?
Is the amoeba is kind of crossing the line there. Isn't the amoeba a tertiary consumer or microbial grazer or both?
They are definitely a microbial grazer. I like this because it doesn't necessarily have primary and tertiary, because like you said, some of these organisms cross that line a little bit. Amoeba are pretty much like nematodes. They will eat anything they can fit inside of them or catch. Any more questions?
Do you want to give a couple of examples for organic substrates and inorganic substrates?
Yeah. So I actually have a whole presentation on feedstocks that I'm going to do a little bit later today. Normally I would do that beforehand, but we will get to that later. I'm going to stick with just the microbes for now, and then we're going to look at some of them. This might cover it a little bit too, but so as Primo was just kind of alluding to, you have to feed your microbes, and they need a good balanced diet or else they're not going to be happy and they're not going to do the things you need them to do in your compost. Bacteria have certain kinds of preferences, as do fungi. So bacteria, as I said before, have really high nitrogen requirements in their bodies. Typically the outside of bacteria's bodies are kind of flimsy as far as soil organisms go. So they don't need a lot of that structural material to consume. They're going to need nitrogen materials. So nitrogen, and you may have covered this yesterday as you built a compost pile, that's going to be your green material, so vegetative plant matter. That's going to be your food scraps. This is going to also include high nitrogen materials. And typically, high nitrogen materials are things that have been pre-processed. Think about manures. We consider this a high nitrogen material. Well, that's because it's already been through the digestive tract of an animal, and a lot of those complex materials have been broken down into simpler ones. The same with, we use a lot of like fermented grains and things like that, and we consider that high nitrogen. Again, yeast and other microbes have already acted on that to break down the really complex structures and provide really simple chemical compounds for soil bacteria to consume. Bacteria have a low C to N ratio. They don't have a lot of carbon needs, so they're really nitrogen fixated. And again, simple structures. So they like materials that are very simple, pre-broken down, sugary substances, things like that. I won't go, you can go deep into these conversations with chemistry. Again, I'm going to spare you that, and we can have side conversations if anyone wants to get into that.
So if we have like, I make my own probiotics at home, so if we have probiotic grains left over, those would be really good for the bacteria, right?
Yes, yes. And it's super important to get that bacterial boom of activity in the very beginning of your compost pile, because that bacteria in later stages is food for those higher organisms. Fungi are on the other end of the spectrum. Fungi have a very high C to N ratio in their bodies. The structural elements of mycelium are very carbon rich. They're very rigid. So fungi need a lot more carbon to create that body structure and create those strands that move out to go find food sources in your pile. Carbon materials. They also like materials that have complex molecular structures. So they're going to break down things like lignin, some of those very persistent materials in the more woody feedstocks that you're going to have, mostly because fungi are capable of creating a whole array of different enzymes that they can excrete to break materials down, whereas bacteria are going to be a lot more limited in the things that they can attack. Usually what I like to think of is size, right? Fungi tend to be a lot bigger, they're a lot stronger. Bacteria are really quite small, and you'll see that once we get under the microscope too. Okay, so there are different stages of your compost pile, and those correlate with different stages of microbial activity. So we have that mesophilic phase in the very beginning, maybe when you first pile your materials together, and then all those materials and moisture come into contact with one another. And what this does is it wakes up all of those microbes. So your feedstocks are one of the delivery systems for getting microbes into your compost pile. It's one of the reasons it's important to know where your feedstocks are coming from and make sure that they haven't had any sort of antimicrobial pre-treatment. So if they have, it's not the end of the world. Microbes can come from other places too. Microbes can come from the rain falling on your pile. They can potentially come from the water source you're using to keep your pile moist, but primarily they come from your feedstocks. A lot of times when those microbes end up in your pile, they might not be active yet. So that's why we're trying to get this perfect balance of ingredients so that they have this incredibly hospitable environment to wake up to, because that's going to encourage them to wake up and start eating. So that's like right in the very beginning. Then we hit this thermophilic phase. So again, 131 to 160 degrees is a pretty harsh environment for most organisms. The only microbes that are going to be super active during your thermophilic phase are typically bacteria. So during that phase, so it's very important that we have this phase. We know what happens during the thermophilic phase. We're killing off a lot of other types of microbes. But what we're also doing is stifling the growth of some of the beneficial microbes that we do ultimately want in our pile. So after the thermophilic phase, we reach our maturation phase. And this is super critical for developing that diversity of organisms that you need for healthy compost and in turn for healthy soil. During the maturation phase of your compost, the temperature is going to come down. Ideally, you're going to stop turning your pile. And this is when the fungi are going to wake up. Fungi do not like high temperatures. They don't like disturbance. So during your thermophilic phase, it's unlikely that you're going to have a lot of fungal activity. They're going to wake up once it cools off, and they're really going to get to work on those woody materials and browns that you've added into your pile to finish the decomposition process. So in the soil, we have this really unique situation, which I think I'm going to cover in like 2 slides. The different populations of fungi and bacteria in your soil actually drive plant succession in the environment. Actually, I do have a different slide for this one. And it's also a nutrient indicator in your mature compost, depending on whether you have a lot of bacteria or a lot of fungi, because the byproducts of their metabolism are going to be slightly different. And they also affect nutrients in different ways as they sit and mature in your compost pile. Okay, any questions? Yes.
Are the fungi active in the mesophilic phase, or pretty much they're dormant until the maturation phase, or they kind of like come and go?
Yeah, so there's a good possibility that there may be active fungi in your feedstocks when you first add them to the pile. Often what happens is the fungal strands get broken up as you turn your piles, as you process your feedstocks, which isn't the end of the world. If the thermophilic phase doesn't kill them, which they are really, really resilient, they will start growing again once the conditions are more appropriate for them. And often what we see a lot of too in our feedstocks is we'll see a lot of fungal spores. So it's very important to have this maturation phase because that's what's going to wake up those spores and show them that there are food sources and a hospitable environment for them to thrive. Yeah.
I have a question that's more on the industrial scale of the community. So I was thinking I know, Prima might have the answer because she did the U.S. Composting Council operators. training, and one of the things I think about is, the industrial scale, and to my knowledge, wants to achieve two results. They want to make sure pathogen reduction and heavy metals aren't present. And when I think about the temps that they're going for, they're doing this in such mass that they're getting those temps really, really high. And then they're churning out a product, and it's going out to industrial agriculture. In your mind, what kind of soil is that making? And what does that look like?
It's creating the exact kind of soil that they are promoting with the agricultural practices they use in those systems. These industries have been working together for quite a long time at this point. And honestly, a lot of agricultural practices would not promote the success of fungi in those soils anyway. So, yeah, it's kind of a catch-22, because it's like you could make the good compost, but depending on how it's being used in the soil and how that system is being treated, ultimately you could negate all of the progress that you made by adding these better materials to your soil based on your practices.
So it's just a totally different frame. Like, they're not even thinking fungal count and fungal health. It's just...
Yeah, by adding fertilizers and different amendments to your soil, typically you're killing off a lot of the microbial life in a lot of different ways. You can disrupt osmotic pressure in the bacterial cells by adding salt products to your soils, calcium, magnesium, lime, things like that. You can disrupt the relationship that the microbes have with the plants that are growing in the soil by adding fertilizers, because you're adding plant available nutrients in huge quantities, which mean the plants don't need to feed the microbes in the soil anymore. And so typically you have a lot of die-off of microorganisms too, because it's a symbiosis that's required. And if that's not happening on one end, then there's no need for those microbes.
It's like a really dysfunctional marriage.
Yeah.
Yes. Yes. Absolutely.
Janel, if you're feeding like high fertilizers like in home gardens, and most people we get that NPK packet from the store and we keep adding it every 2 weeks, are we getting those microbes high?
Honestly,
are we overdosing them, or is there a point no, I'm done.
yeah, I mean, typically what that does is those materials are salts. They're ionic, so they have a charge. And yeah, you can completely just overload your soil. So there are different functions microbes serve, and you're surpassing those functions, essentially, and potentially causing direct harm. Because if you have a high salt concentration here and not inside the cell, it'll suck water out of those microbes out into the soil because of the salinity difference, the difference in concentration of ions in those solutions. So yeah, if you don't just directly kill them, you're definitely disrupting their life cycle and their function altogether in the soil. Definitely. Not good. Organic matter is the best fertilizer. Okay, so we've been talking a lot about good microbes. There are some questionable microbes and some ones that we would definitely consider bad. And they have different lifestyles. So I talked about this in the beginning. When you're looking at your pile breathing and you're experiencing that, sometimes what the microbes are breathing out doesn't smell super great. And that's an indicator that we have a different type of microbe acting on our piles, and there are going to be some consequences of that. Not all bad. A lot of us are familiar with fermentation, and that's lovely, especially if you like alcohol. However, alcohol in the soil isn't ideal, and there are some even worse compounds that can be created. So we have different types of organisms, aerobes and anaerobes. Aerobes like oxygen, anaerobes don't. Aerobes prefer oxygen-rich environments. This is something that we're attempting to maintain in our compost piles with aeration tubes or turning. Rapid decomposition. So this is one of the biggest benefits of aerobic composting, is that you are going to get breakdown at a very rapid speed. The type of metabolism these organisms have, they have a lot of competition. A lot of microbes and macroorganisms in the soil are aerobes. So everyone really just has to get after those nutrients while they're available. This happens very quickly. They breathe out carbon dioxide. Maybe that's not super great, but we're going to talk in a little bit about how they are also sequestering carbon in the soil and replenishing that pool so that the entire cycle can take place. Breathing out CO2 isn't the end of the world as long as we're completing the entire cycle. Aerobes create soil-stable nitrogen. So this is going to be your plant-available nutrients, nitrogen specifically. And bacteria are really the drivers of this process in your soil. It's a hot process. Again, there are benefits to having thermal composting, thermal and aerobic composting. Thermal composting can help it's gonna help kill pathogens, which is great. It's gonna help kill weed seeds, materials that we don't want sprouting back up after we put our compost out. And it's also going to help in the process of breaking down certain pollutants that may end up in our compost piles. So we consider aerobes beneficial organisms when we talk specifically about the compost environment. Anaerobes have a slightly different life cycle. They prefer low oxygen. There's two different types of anaerobes. And there's one specific class of anaerobes that are a great indicator of where our compost is headed. Rarely do we see obligate anaerobes in compost. It's really challenging to make your compost pile go completely anaerobic, it turns out. Usually we see that in things like sewage sludge, swamps, wetlands, things like that, where there are places in the muck where there's really almost no oxygen. So we have facultative anaerobes, which means they can survive with some oxygen, but they prefer lower oxygen environments. And these are indicator species when we get into microscopy that are going to show us that the oxygen in our pile is on the downslope and we need to do something about it. Anaerobes are also decomposers, but they act very, very slowly. Their life cycle processes are not super efficient. There's lots of different byproducts, and it's just a very slow, typically cold process. So again, if you go out in your pile, maybe there's some fumes and gases, maybe it's a little stinky. If you touch that pile and it's cold, your decomposition is not happening aerobically. These are some of the lovely gases that we get with anaerobic decomposition, and this is where the smells come from. Bad smells in your compost are going to attract pests because it's indicating to them that there is still a food source available, versus thermal composting, where when this happens quickly and it's only CO2, nothing is being attracted to your pile because there's no indication that there's still a food source there. Methane, we're all pretty familiar with, I'm sure, one of the worst greenhouse gases that gets emitted from different types of industry and agriculture. Real stinky. There is some carbon dioxide still, again, facultative anaerobes, not full anaerobes. And hydrogen sulfide, all lovely caustic materials that are going to come out of an anaerobic pile. So as well as gases, anaerobes are also going to create other byproducts that are going to go out into your compost and potentially into your soil. These materials can inhibit germination of your plants in your soil and even directly cause damage to those plants if they come in contact with large amounts of them. Alcohols, strong organic acids, acetic acids, things like this just really aren't great for your soil. And you're going to lose your nitrogen. So one of the other gases that's emitted during anaerobic decomposition is ammonia. Horrible smell, and that's your nitrogen. leaving in gas form from your piles. So there are certain forms of anaerobic composting that people employ, but they select very specific organisms to do that work. So we're not going to say all anaerobes are bad, because that's not true, and they exist because they serve a function in the environment. But in thermal aerobic composting, we're definitely aiming not to have anaerobes present. I'm running out of time. Any questions? So I'm going to skim this fairly quickly, because I know we're trying to move on, but we also started a little bit late. So fungi and bacteria, you're going to see this information if you get your compost tested, presented as a ratio, a fungal to bacterial ratio. And what that tells us when we're talking about the quality of our compost is what that compost is going to be good to be used for. So we were talking about, heavily managed agricultural soils. Typically, these environments are going to have something like a 1-to-1 or 2-to-1 fungal to bacterial ratio. The fungi you're going to see present in these places might be some mycorrhiza. There are plants present in this system, and most agricultural plants require mycorrhiza to pull nutrients from the soil, and they have an exchange. What you're going to be missing in these soils are some other types of fungi, which are saprophytes, another term you may be familiar with. These are going to be the mushrooms that you see out in the forest, typically when you dig in the leaf litter and see all that white mycelium growing. They break down woody materials. So you're likely going to be missing those. They do not appreciate the disturbance in heavily managed agricultural systems. So compared to ag soils, if you go out into the forest, specifically coniferous forests, you might get a fungal to bacterial ratio of 100 to 1,000 to 1. These are fungally dominated systems. The trees don't require heavy inputs of material of nitrogen, which, like we said, that's what bacteria handle, right? They are really the nitrogen cyclers in your soil. So trees have a ton of nutrients stored in their bodies because they're a huge organism, whereas an annual vegetable plant needs to grow fast. It may have only a few months to completely mature, create fruit, seed out, and die. So it really requires this fast, heavy turnover of nitrogen, whereas forests don't have the same needs. So something to consider if you have that test done for your compost and what the best applications of your specific material might be. This is how microbes cycle nitrogen. So I was really just going to cover a couple terms in here that you might not be familiar with. So nitrogen fixation. Microbes have the ability to take atmospheric nitrogen and fix it into a plant-available form in your soil, which is incredible. Some of these nitrogen-fixing bacteria have to live in an anaerobic environment to be able to do that. These are not aerobic microbes. They are not breathing in oxygen and breathing out CO2. Clearly, because we're getting nitrogen out of this. So again, those plant associations when we're talking about soil are very important. There are also some free-living microbes that live in the soil that can accomplish this too, typically at a slower rate. So nitrification is going to be this conversion of different types of nitrogen, and I talked about that a little bit earlier. So in your compost process, these really complex organic materials are going to be broken down typically into ammonium, and then from ammonium it's going to be turned into nitrates. You might be familiar with nitrates. All of the fertilizers that you're going to buy are, any nitrogen fertilizers anyway, are all going to be in the form of nitrate. So you might wonder, why is it important to have ammonium then? ammonium is actually a very stable, non-leachable form of nitrogen in your soil. Nitrates, on the other hand, while they're great because they're immediately available to your plants, also will leach away with water, over-irrigation, rain. You're going to lose that nitrogen very quickly. So what happens is this ammonium sits as a pool of nitrogen in the soil. The plant talks to the microbes and requests nitrogen, and the microbes convert ammonia to nitrates. So it's a much more stable cycle versus dumping a bunch of nitrates in the beginning. None of these nitrifying bacteria are going to be required anymore. So when that nitrate is gone, you don't have a pool of ammonium in your soil for the microbes to cycle to your plants anymore. Denitrification. This is what's going to happen to your stable and plant available nitrogen in your soil when you have anaerobic processes going on, and that's going to leave as ammonia. And plant available nitrogen, well, we covered that. That's going to be ammonium and nitrates. And plants actually do use ammonium as well, to a degree. Oh, no. Carbon. So this is one of the most exciting things that we talk about when we talk about soil, and this is definitely something that Cal Recycle and CCI is very interested in us talking about when it comes to soil, and that's carbon. So this is a very complicated chart of what actually happens in a healthy ecosystem with carbon. So plants, as we all know, they're going to take in carbon dioxide from the atmosphere, and they hook that onto these big complex sugar molecules. It turns out that most of the carbohydrates that these plants are making and expending all that energy to create, they are feeding to the soil microbes. I think some to the tune of like 80% of these sugars they create are not for their own metabolism. So what they do is they put out exudates, fancy word for these sugary carbon substances, carbohydrates, and they feed it out to all of these microbes in the soil. And in exchange, these microbes go and collect other materials from the soil. They use their enzymes to break down these materials and bring them to the plant. So fungi does that by actually connecting to the plant roots and then spreading out in this vast network in the soil and collecting materials from different places. Microbes can also change the pH in certain areas to create the release of different types of materials in the soil for the plant. Bacteria have a slightly more aggressive relationship with plants. So it used to be believed that plants got these nutrients from the soil just by diffusion. So the nutrients exist in the water around the plant roots, and based on concentrations on either side, the plant roots were able to suck in some of these nutrients, which just is not true. So mycelium, they have a certain relationship. Bacteria can actually be taken inside of the plant root. The plants can dissolve the outer layer of the cell, suck the nutrients out of the bacteria, and then spit them back out into the soil with a new outer coating to go search for more nutrients for them. So I thought that was pretty cool. It's called rhizophagy, and there's actually some really interesting videos that were taken, I think it's Rutgers University. Real exciting. So anyway, the plant will have interactions with these different microbes, and there's a lot of different ways that the carbon remains in the soil after these interactions. So microbial biomass, as we said before, fungi create a ton of carbon-rich structures in their mycelium to keep them safe, to create these tunnels for them to move through to go seek out nutrients in the soil. So that's one of the stable carbon pools that exists. So encouraging fungal growth is critical to that. There's also some materials that fungi create to kind of glue soil particles together to attach themselves to different food sources in the soil called glomalin, which is, I've heard the term before, it's still kind of a new topic to me, but apparently researchers are finding that this substance created by fungi is another stable pool of carbon in the soil and makes up something like 70% of the carbon materials in the carbon pool in the soil. So that's exciting, as long as it doesn't become an industry pitch. Yeah, and then there's another way that these microorganisms are creating stable organic carbon in the soil. And that's by creating humic substances. So I'm going to keep this sample for the rest of my life, I think. This came from our soil retreat in Sonora. And it's just to demonstrate what I'm about to talk about. So humic substances, these are going to be the skeletons, these carbon-rich, super resilient skeletons of the materials that are being broken down by these organisms. So once these organisms have stripped all the different materials off of your leaf litter, off of your woody debris, off of your plant materials you're putting into your compost, there's these carbon skeletons that are left. And these are going to connect to other bits of carbon materials that are left in your soils and create these huge molecules, which we call humic substances. There's different types. There's a different stability related to each different type. I'm not going to get into super details. But these substances are responsible for the water dynamics that happen in our compost piles. They are responsible for helping with aggregation of particles in our soils, which creates texture and structure. And they're also responsible for nutrient exchange. So these huge complex molecules are going to have all these different charged sites all across their surfaces. And those sites are going to attract different molecules and ions in the soil. They're going to hold on to them, and we call those exchangeable nutrients. So when a plant needs something, the microbes are going to go to this molecule, find the specific ion or nutrient it needs, pull it off, and go feed it to the plant. And then that opens up that charged site again for something else to connect to it. So they really create a lot of the dynamics in the soil. And there's a super easy visual way to tell if you are creating humic substances in your soil. I shook it up. I shouldn't have shook it. So if you want to pass this around. So there's this very distinct color that you're going to get when you're starting to create humic substances in your compost. And this is going to happen during the maturation phase. So this is one of the most critical reasons to let your compost sit for that up to 6-month period, is because you're really not going to get your materials broken down to that point until you reach that time frame. So some of you may know that there's talk about what finished compost looks like, and it's that 70% dark chocolate, kind of chocolatey brown color. That's the humic substances. That is the color you should see in water when you put a little sample of your compost in it and shake it up. And you can do this test. You'll notice a difference. Some of us have really heavy clay soils, which also might create that kind of milky brown appearance, but this is extremely distinct, and I like to pass this sample around so you can really see that. That sample is from 8-month-old compost. And actually, when we looked at different samples during that retreat, you could tell the difference in the age of the samples just by looking at them like that in the test tube.
So darker the better.
To a certain extent. You don't want it to be black. Black can imply that you have anaerobic processes going on, and you're ending up with a different end product. So, I'm almost done. Anyone have a question, though?
When you said black, Janel, is it color of water or the compost color?
So that brown you're seeing is the materials in the compost, these humic substances in your compost. They're not actually water soluble, they're just suspended in the water. Important distinction. You need acids to dissolve those. Yeah.
And this is gonna seem really basic, but
That's
everybody just describe with what's going on under the soil and the plants absorbing nutrients. I was talking to Frida last night about growing vegetables, and she said she thinks that vegetables grown in the soil taste better than hydroponics. What's your take on that?
Yeah, well, it's a very complex topic and a very complex question, because plants that are in soil are likely to have a lot of different experiences. Even to the, we even talk about, when it comes to soil, it's different than compost, and there are different things happening with the microbes in the soil. Like, you need a certain percentage of bad microorganisms to be able to kind of prompt your plants to do certain things. So like, there's these certain stress factors that are actually improving the health of your plants when you're in soil, things that might not be happening in a hydroponic situation. Also, in a lot of hydroponic situations, it just goes back to you're using all of these inorganic fertilizers. It's a bunch of salts, so you're fulfilling the very basic needs of your plants, but not all of them. It's just like us eating processed food. Does it cover your very basic needs? Sure. Can you survive off of it? Sure. But are you thriving? Unlikely, because you're not getting this diversity of nutrients when you need them, like a plant would in a soil situation, particularly in a microbially rich, healthy soil situation. So yes, it's a very complex topic, and actually nutrient content of produce based on different soil practices is a really hot ticket research item right now too. My personal opinion would be yes, soil is better, but that's just me.
It's complex and we need more.
Yeah, it's very complex. I know, I was like, how do I keep this really quick and simple? And I don't know if I can. Okay, so another really exciting thing about microbes in your compost is they are going to help deal with some really gnarly stuff that we might end up with. Not going to get deep into this. I have stacks of research papers if anyone wants to go down like a super nerd rabbit hole, but I'm not going to drag you into this because it's 8:30 in the morning. But suffice to say, there is evidence that microbes can deal with all of these things in your compost, and in turn, by putting those microbes back into the soil, it's likely that we can speed up the process of breaking down these materials in soil environments as